Hello, my name is Ellen Goza. I worked with Dr. Michelle Guidone of the Biology Department at Georgia Southern University Armstrong campus on this ongoing research project. We are interested in magnolia trees native to the southeastern United States. They grow naturally in coastal forests and are often planted as ornamentals. Our project was focused on observing a specific insect pest that is often found on magnolias, called the false oleander scale. Once the scale attaches itself to a leaf, it remains there, living off the sap of the leaf until it dies. We wanted to investigate the potential for differences in scale infestations between a semi-natural habitat where the trees grow without having been planted versus cultivated trees in a landscaped setting. We selected multiple trees on the Armstrong campus that represented each habitat. Leaves from each tree were photographed at the source tree. Photos were later analyzed in a program called ImageJ. We measured several characteristics for comparison, which were surface area, perimeter, and length of leaves, as well as total scale abundance per each leaf and scales per centimeter squared. A summary of these measurements is depicted in the graphs on the top right of the poster. We also estimated the percentage of each tree's leaves that were covered in scale insects. To estimate, we created categories describing the density of scale on the leaves. These include none, sparse, moderate, and abundant scale coverage. Dr. Guidone, another research student, and I evaluated each tree's percentage estimated to the nearest 5% for each category. These three independent estimates were averaged for analysis. Based on the percent coverage estimates, a pattern emerged. Trees in the semi-natural habitat exhibited a greater percentage of scale coverage than those in the landscaped setting. We also observed differences in leaf size, with landscaped tree leaves having a smaller perimeter and surface area. While we initially expected trees in the semi-natural setting to be more resilient and therefore less infested by scale, we were surprised to realize that this is not the case. Contrary to our initial hypothesis, the trees in the landscaped setting exhibited lower levels of infestation. The differences observed in leaf size may be due to genetic or environmental factors, which could influence scale infestation. Further examination of these and other factors is required to determine the cause for the difference in scale infestations across habitat types.